Good morning, good afternoon, and good evening. Thank you all for attending today's webinar titled Resolving Flexibility and Heterogeneity Analysis with 3D Variability Analysis. This webinar is organized by Structura Biotechnology, the team that builds and supports the cryoSPARC software package for single particle cryo-EM. Today is the first in a series of webinars that we're hoping to offer on various topics related to cryoSPARC and cryo-EM image analysis. A few housekeeping notes about today's webinar. The webinar will be recorded and the recording will be made available to view after the program. We will also have a question period towards the end of the hour, so please submit your questions through the Q&A feature, not through the chat, which will not be moderated. And with that, I'd like to turn it over to our presenter, Ali Punjani.
Hi everyone, my name is Ali. Thank you so much for joining us today. We're excited to be presenting this webinar about 3D variability analysis. The plan for today is to talk about 3D variability analysis in depth, explaining the intuition behind the algorithm and how the algorithm itself works, some mathematical details and properties, how it can be used to resolve flexibility and heterogeneity from single particle EM data, as well as results on interesting data sets, practical considerations for when you're processing data of your own, and hopefully some engaging Q&A. To get started, I'd just like to give a tiny bit of background about cryoSPARC itself for those who are not familiar with it or who haven't already started using it. It is a full pipeline computational platform for single particle cryo-EM. It includes computational tools for all phases of single particle data processing and allows users to interact with their data intuitively, processing the data in a visual manner and in a streamlined user interface. And today it's used around the world on a daily basis by thousands of users at hundreds of institutions and with many, many citations of major structural studies in the last few years. It's now one of the most popular tools for single particle EM and the fastest growing in adoption, which we're very excited about seeing. It was originally started as a research project at the University of Toronto in the computer science department. And the first versions were built in 2016. And since then, it's been under heavy development by our growing and excellent team that we've assembled at Structura Biotechnology, Inc., which was spun out of the university specifically to build and support cryoSPARC. And our focus is to build cryoSPARC and to combine the new algorithms that we developed, like the one that we'll talk about today, 3D variability analysis, along with fast GPU implementations of various methods that we optimize through an engineering effort and professional software design to create a reliable, robust, and high-performance computational platform. We see single particle EM continuing to grow in adoption and capabilities in many biological contexts, and our goal is to support the cryo-EM community with the best possible tools and workflows that we can provide, which is why our team spends a lot of time and energy continually improving cryoSPARC and distributing it freely for academic users, and it's also available for commercial use through our company. So I'm very happy today to be talking to you about 3D variability analysis, which is an exciting research project that has been ongoing for some time, since early 2019. It was presented last year at the 3D Electron Microscopy Gordon Conference, and it has since then been released in cryoSPARC version 2.9 actually at that time, and many groups have already been experimenting with it and using it. Recently, we had a chance to get out a preprint describing the method, as well as the algorithm, the results in more detail, along with some tutorials for how to use it in practice. Today's presentation will cover all of that in hopefully a good amount of detail. We will begin from the fundamentals, but I am assuming that the audience has some familiarity with single particle EM reconstruction, generally speaking. And I'd invite anyone who wants to read more in depth to check out the preprint. Thank you. So diving right in single particle EM, all computational methods and algorithms are designed in the context of the physical image formation model of the microscope and the sample that generate the images that we see on the camera. That image formation model tells us or helps us to predict what kind of images we should see given a particular structure, and also allows us to do the inverse, which is to infer the structure and its properties from the images that we see. There are two parts to the image formation model that are very important: the microscope and the sample itself. The microscope is, most people are very familiar with in terms of how it overall works. There's a few steps. The particles themselves are frozen in ideally a random and unknown pose. So that's 3 dimensions of orientation pose that we don't know, and 2 dimensions of in-plane shift within a particular image for every single particle protein. That protein is imaged in a transmission electron microscope to generate 2D projections on the camera, and it's imaged at a low dose to limit beam damage, which creates a high level of noise. The images themselves are also corrupted by a microscope contrast transfer function that depends on defocus and alignment of the microscope, and that may even be different from particle to particle. The particle images themselves that come out of this ideally are very clean, and we can see individual proteins and hopefully purify them down to remove junk and other non-particle images. And at the end of the day, what we will be left with is a set, usually tens of thousands or hundreds of thousands and millions of particle images that make it possible for us to carry out the task of reconstructing a 3D structure of the protein target that is in the sample. In general, in single particle EM, reconstruction of the target is the task. However, in 3D variability and when resolving heterogeneity, it's really the dynamics, the motion, the heterogeneity, association, dissociation, and conformational changes of the protein that we're interested in. It's possible to investigate that using single particle EM thanks to the characteristics of the protein itself, which are the other part of the image formation model. The protein itself may exist in multiple conformational states. There may even be distinct particles in the sample. And what's most important is that there may be flexibility or disorder in a single protein that may be present in the sample. Because the protein is going to occupy multiple conformational states throughout its possible landscape of states that it can take on in the population of images that we collect, there's a chance that we may sample enough images to actually be able to derive some insight about the conformational landscape of the protein. And that's really the hope of all heterogeneity analysis techniques, including 3D variability analysis. From a computational and statistical perspective, all of these data characteristics are dealt with by formulating the reconstruction problem in the heterogeneous case or the homogeneous case as a statistical inference problem. I won't go into too much depth into the probabilistic formulation, but I do want to explain how the reconstruction task is set up and cast as a mathematical optimization problem, since that's the core of all methods, including 3D variability analysis, and it makes it generally a little bit easier to understand what is going on. So the diagram on the right is what's known as a probabilistic graphical model. It connects all the parts of our problem, each circle representing a variable that we may or may not know or may not observe, connects them with arrows that tell us how information flows between variables. So for example, X is a particular 2D image that we observed in the microscope. C are the corresponding microscope settings, contrast, transfer function, and other settings for that image. Phi are the poses that we don't know for that particular image. We don't know which way we're looking at the object in that image. And in the simplest homogeneous reconstruction case, V is a 3D structure that explains all of the images. The red arrow is the most important arrow. It captures the image formation model, which we just talked about, which is the relationship between the 3D structure itself and the images that we observe in the microscope. In general, what we're trying to do in this scenario is work backwards through this arrow so that given the images and the CTFs, we can infer and then ignore the poses, which we don't really care about, and infer the 3D structure by working backwards through this arrow so that we can figure out what structure best explains the data. The mathematical object that we'll be dealing with is this probability function. It's the probability of a particular 3D structure V given all the images that were collected in the microscope. Each 3D structure has a different probability given the images, and the one structure that has the most probability, the highest probability, is our most likely maximum likelihood structure. And that's what we want to infer by solving an optimization problem that's taking the maximum over possible 3D structures to find the one 3D structure that has the highest likelihood. In computer science and machine learning, optimization theory and algorithms are extensively studied for all types of scenarios and all types of problem setups. And this basic version that I just showed right now is what the problem setup for the homogeneous reconstruction. We're going to extend it a little bit to come up with 3D variability, which is a modified and extended version of this same overall setup. Just making this a little more concrete, in the standard homogeneous model, we can write down our image formation model in a simple way. And this should be familiar to most people who've done single particle reconstruction before. What we have is a single 3D volume, V, that is projected by some pose, phi, and that the result of that is a 2D image that's corrupted by the CTF of a particular image X, and then noise is added to that, and that's what generates a particular image X. That's this forward arrow. And what we want to do is go backward through that to infer V from a whole bunch of measurements of X. In this simple case, the optimization problem that we have to solve, there are standard algorithms which are now mainstream in single particle EM that allow us to solve that optimization. For example, if we're starting from scratch without an initial model, we can use Ab-initio reconstruction in cryo-SPARC that's based on the stochastic gradient descent algorithm. We can also use iterative refinement that's been used for decades to refine 3D structures from cryo-EM data to higher resolution once we have an initial model, both of those algorithms trying to solve the same optimization problem. We can also extend this simple model to the slightly more complicated case of heterogeneous structures. So in that case, instead of having a single 3D volume, we can extend the model to having multiple distinct different 3D volumes along with a new set of latent variables. And we can try to infer all of the different structures that are present, discrete structures that are present in the data set, along with the classification of particles into each structure. All of these techniques, homogeneous refinement, heterogeneous refinement, ab-initio reconstruction, etc., are already mainstream in use for single particle cryo-EM today. And they can get us so far in trying to resolve heterogeneity. With this context in mind, let's zoom out a little bit and consider the problem of conformational heterogeneity intuitively. If you think of all the images that are collected for a given data set, for a given protein in the microscope, each single particle image, one little square, those images collectively occupy a space. They occupy positions in a space of all possible particle images that could have been collected. That space of all particle images is this sort of diagram here. I'm just showing 2 dimensions, even though the space actually has tens of thousands of dimensions, depending on the number of pixels in an image. In that space, each image can be a dot. Each dot corresponds to a single particle image. And what we hope intuitively is in that space of all particle images, if we had some discrete heterogeneity, we would see the particle images form clusters. The particle images should be that are similar from the same 3D structure should be grouped together nearby to each other in this space. And what we hope is that in a discrete classification scenario where we have multiple different, let's say, complexes or partially assembled versions of a complex, etc., where there really is just discrete heterogeneity and nothing in between, we would see these clearly separated clusters. Of course, there's noise. Every particle image is moved around by noise. There's a contrast transfer function. Each particle image is actually only a single view of a 3D structure. And so even 2 images of the same 3D structure may not look the same because they're from different viewing directions. But abstracting that all away, we can think of images as dots in this space. So in the discrete case, we would expect clusters. In the continuous space, in the continuous case where we have a protein that has maybe a flexible region, we might expect all the particle images to lie in a manifold, what's called a manifold, a multi-dimensional surface in that space where the particles are all grouped together and have some directions along which they vary. One of those directions might correspond to a certain kind of flexibility in the molecule. This is kind of what our intuitive picture of conformational heterogeneity of both kinds should be. Unfortunately, in practice, we can't actually interact or visualize or inspect this space of all particles. Even though the space and the arrangement of particle images in that space contain all the biological and functional information about protein dynamics that we're interested in, we can't actually look at it. And so we have to resort to techniques that we have at hand, which are essentially classification-based methods to try and separate the space into regions and hope that each region corresponds to something useful against the underlying geometry that we're really looking for. And some questions arise in that process. For example, on the discrete side, if we can't see the space, how do we know how many classes there are? How many different clusters are there? Or how separate are there? Are there maybe some large clusters that are divided into subclusters? We can't really see that or know that ahead of time. Similarly, on the continuous side, we can't know if we have multiple degrees of freedom. Are there different ways in which the object can change? Is there only one degree of freedom in the protein? We also don't know what the types of flexibility actually are. Is it bending? Is it twisting? Where are the hinges? Where are the joints? Is it even rigid motion or is it fully flexible bending? We don't know any of those things ahead of time. And so with the tools that we have at hand so far, we can't really work perfectly well in either of these scenarios. For example, if we start to think of what would happen if we use standard 3D classification or heterogeneous refinement techniques in either of these scenarios, in both scenarios, we're likely to not do very well. On the discrete side, we would hope that if we run, let's say, 3D classification with 3 classes, we might find these 3 clusters. But in practice, we know from experience, and anyone who's tried doing heterogeneous classification knows that every time you run classification, you may get a different result. Sometimes you might find that you only got 2 classes, and one of them is, the third class is empty. And one of the classes is actually an average of 2 different conformations. So they weren't actually classified or separated. Sometimes we may find that one cluster is missed entirely and we end up with two clusters that split, that have the same structure and split one conformation. Neither of these is really helpful, and we can never really know, we can never really verify whether our clustering has actually captured all the variability in the data set. Similarly, on the continuous side, we would hope at best that our discrete clustering technique would provide us with clusters that span the variability of our protein. But in practice, this is almost never the case. It's much more likely that our clustering algorithm, just based on how it's initialized, will converge to a situation like this, where we end up with one large cluster that, again, just averages together all the particles and doesn't actually show us any of the continuous heterogeneity, and some empty clusters. So in both of these scenarios, the idea of discrete clustering doesn't get us all the way to the answer. And this is where 3D variability and techniques like it try to step outside of the box. 3D variability is based on a completely different, fundamentally different model from classification. There are no discrete clusters in 3D variability analysis. The actual model is based on what we call a linear subspace model. And it's fundamentally different in the sense that it's a continuous, multi-dimensional family of 3D structures that we're trying to find. We're trying to find this family that explains all the data together. Instead of discrete 3D structures that don't have anything to do with each other, we're looking for a continuous family that's all related that explains all of the data. And the family itself is very simple. It's a linear subspace. So that means the family is defined by a mean, which is the center point of this cross in both cases, and some variability components or principal directions. There are many names for these types of things. And this whole idea is not new. It's been explored by many people and many groups, but it's substantially more difficult to work with than the previous techniques that we've described so far. So in 3D variability analysis, in the linear subspace model, the linear subspace itself is characterized by a mean, which is a single 3D structure that maybe is at the center of all of the particle images, and some variability components that show us in which directions in our space of all particles is there significant variability amongst the particles. If we really had a perfectly homogeneous set of particle images, we would expect that all of them are the same within the noise that we expect from the microscope and the low dose. Other than that, there should be no difference between particle images, for example, from the same viewing direction. When we do see differences between particle images, that variability is what we're trying to capture with a linear subspace model. And like I said, this is not a new idea, and many people have tried to work with it, but because of the nature of the problem and the mathematics behind it, it's much more difficult to actually solve. Diving in a little more and connecting this with the probabilistic setup that we saw earlier. In our probabilistic setup, we have images that we observed, CTFs, poses that are unknown. For now, we're going to assume that the poses are known. We're going to assume that before we start using 3D variability analysis, we've already done a consensus refinement of all the particles that we have. So we have a single 3D structure, that's the average of everything, the mean, and we have poses for every particle image that are fixed relative to that mean structure. The model that we are actually going to use for 3D variability analysis looks like this. Instead of a single 3D structure that gets projected and then corrupted by the CTF, we have a family of 3D volumes, density volumes. And that family is indexed by some per particle coordinates, Z. The per particle coordinates tell us which member of the family is present in a given single particle image. Each single particle image only has one protein structure in it. The family contains or captures the variability amongst all of the different versions of that protein that may exist. So that may be a bending flexibility, or maybe a particular subunit disappearing or reappearing. Across that family, each single particle image contains or has within it only a single protein, a single conformation. And Z, the per particle coordinate, tells us which member of the family it is. The Zs are continuous since the family is continuous. So having Z values greater or smaller lets us choose a particular version of the structure. And once we take the family and apply Z, we get a single 3D structure again, which is one member of the family. We can project it by the same pose, corrupt by the CTF, and now this time also scale by a per particle scale factor, and that's to capture variability that we don't want to capture in our families, things like contrast changes because of ice thickness or other factors, plus noise. And that's the entire model for 3D variability. Now, this family of volumes itself is represented in a simple linear way, which is a sum of parts. The first part is a static part, and that's our mean density, the basic 3D structure that we already refined using all the particle images, the mean of all those particle images in 3D. And that's fixed and known ahead of time. The other part of the family is a sum of variability components. Each variability component, now Vk, where k goes from 1 to capital K, each of those V's, the variability components, is another 3D volume that can modify the basic 3D volume. And the modification that's applied depends on what the values inside of the variability component are, positive or negative values, times the per particle latent coordinate. So the per particle latent coordinate tells us how much of each variability component, all k of them, to apply to the basic structure to generate the structure that's present in the given image X. That's what the linear subspace model says. And then everything else is the same. The only other small detail is that we enforce or we request that all of the variability components be orthogonal to each other so that they don't end up representing the same variability. That's the entire linear subspace model for 3D variability analysis. And what it does is it naturally captures a model of a continuous family, as I've said, of 3D structures that try to fit the data. The hard part, once we get to this point, is figuring out how do we actually fit this model? How do we perform that inference optimization to fit this model to the data? The parameters we have to fit that we don't know are all of the VKs, we don't know the variability components. The Zs, we don't know the position of each particle along each variability component, and we don't know the alphas, the scale factors. So we have to fit all of those at the same time to the particle images. And that turns out to be very difficult for a few reasons. And many people who have worked on this problem have come up against these difficulties and tried to solve them in different ways. There's a lot of literature in this area. Some of the earliest techniques started with an idea of bootstrapping, a statistical technique, and there's a lot more detail in our bioRxiv preprint for those who are looking for more literature about different ways of solving this problem. But all of those techniques came up against these three roadblocks. One, each single particle image that we observe, X, is only a single observation from a particular direction. And so it's only a partial observation of the variability components. We never get to see a whole 3D volume from a single particle image. We only get to see a particular direction. And so that's one thing that makes the problem difficult. The second thing is the dimensionality of the problem. As an optimization problem, the number of parameters is huge. There's so many voxels in each VK, and there's so many Z values that we have to optimize over. And the last thing is that each single particle image is corrupt by potentially a different contrast transfer function. If that wasn't the case, then this model might be a lot simpler, and we could use standard kind of off-the-shelf methods like principal components analysis that tries to solve a similar problem in the case where all the data is generated in exactly the same way. In our case, because of the CTF, the images are not generated in the same way, and so standard methods don't work. It's within this context that we started developing and working on the ideas behind 3D variability analysis. 3D variability analysis tries to solve this exact same problem. And the main technical advance in it is just the development of a new inference algorithm that allows us to actually solve the optimization problem and fit this model to the data to high resolution using all the particle images at the same time, correctly accounting for the CTF, and without making any approximations. The actual algorithm itself is related to probabilistic PCA and the expectation maximization technique for this kind of inference, and all the details are in the preprint. What we were able to do is derive a direct solution for that inference algorithm and create a fast GPU implementation of it, since it's a relatively heavy operation. And what this all means, the big news, is that this technique now, compared to all the other methods that have existed in the past to try and solve this linear subspace model problem, 3D variability is the first method that can actually resolve high resolution motion, even of small particles with multiple different variability components at the same time, and really show us the flexible and discrete heterogeneity within a sample in a really straightforward and new way compared to mainstream techniques like 3D classification. So that's what 3D variability analysis is all about in a nutshell. In terms of actually understanding the results when you use it, its purpose is to help us resolve flexibility and conformational change. What actually comes out of the algorithm are two things. One, the variability components themselves, which, as I said, are entire 3D volumes, the same size as the basic 3D density, which is V0. The variability components have positive and negative values, and they tell us for the particular type of variability or molecular change that is being captured by this variability component, where do we need to add or subtract density in the basic 3D structure to create different versions of the protein molecule. The second thing that comes out of the algorithm are the latent coordinates for each individual particle. And those indicate, as I said, the per particle position along each variability component. So if you have 5 variability components, k equals 5, then you will have 5 per particle latent coordinate numbers for every single image. And those per particle latent values can be plotted on a histogram. They provide us with a distribution. They show us where do the particles lie along some dimension, which is the variability component. So this is a simple example on the left here, a ribosome, 2D slices through a ribosome protein where we've done 3D variability analysis with just one component. We have the basic 3D density here in black, the variability component with red and blue showing positive and negative values. This one actually happens to correspond to, I believe, rotation of the head region of the small subunit. And then we see for that same variability component, where are the particle images along that variability component? So some particle images have a strongly negative value, meaning that for that particle image, we need to subtract whatever the variability component says times whatever value the latent coordinate is. And some have a strongly positive value. That means we have to add. With that information, we can start to construct, we can start to actually inspect what kind of heterogeneity there is in a data set. One important thing to remember when you're using 3D variability analysis is that the algorithm itself doesn't have any assumptions baked in about what causes the variability. It could be motion, it could be flexibility, it could be a dissociation of the subunit or falling apart of a complex, it could be ligand binding, it could be just as simple as junk floating above and below the particle in the ice that causes variability between particle images. Anything that causes variability between particle images can be picked up by 3D variability analysis. We'll talk about what's detectable and what's not in a few minutes, but this is important to keep in mind. Motion, however, is a special case since a lot of times what we're interested in is the dynamics, the flexible motion of a molecule. And so I'll just talk for a minute about how 3D variability analysis models motion. Motion is not exactly linear. So the linear subspace model isn't a perfect representation of moving objects. However, in certain cases, which are very common, the linear subspace model is a very good approximation to motion. In these images, what you're seeing in the top row here are 2D images, synthetic data that we've generated so that we can do 2D variability analysis, just as a toy example. 2D images where this black dot is moving from a top position to a bottom position. The two positions are shown here superimposed. So that's how far the dot is moving from one end to the other of the spectrum. And in the bottom row, you're seeing the results of 2D variability analysis on this data. What we're seeing here in the variability component is some positive and some negative density. And that shows us where density is going to move around as we move from one end to the other of the spectrum. And the bottom row of images here shows us what the variability analysis linear subspace model generates as images at these same latent coordinate positions. And we can see that it's a very faithful approximation of the motion of the black dot. When we look at a different scenario where we now have a smaller object moving the same distance, we can now see that the variability component is different, but the approximation to the motion is much worse. And that's really the crux of the matter. The linear subspace model and 3D variability analysis are a good approximation to motion when the motion distance is a similar distance to the size, relative to the size of the object. So in the case of the first two black dots, we have a motion distance that's about the same size as the object itself. You could also think of it as the resolution of the object. And in the second case, we have a motion that's much larger than the object. And those are the two cases where 3D variability works very well to capture motion and where it doesn't. We'll come back to this as well in terms of what to do in practice when you're working on a real data set. Next, what I'm going to show is just more example results on the same ADS ribosome data set, now with 4 variability components. So we can get a feel for what the variability components look like, what the motion looks like, and how the latent coordinates of particles are spread out. Then we'll talk a little bit more about the properties of the algorithm and how it works in practice. This image is showing in these 3 scatter plots after running 3D variability analysis on a data set of 100,000 ribosome particle images from a public repository data set. We can see that the 3D variability analysis algorithm has captured variability along 4 different components. We asked for k = 4, and so we have each dot on these scatter plots being 1 particle image laid out along the 4 different components. We're just showing scatter plots of adjacent or sequential coordinates so that we can visualize this 4-dimensional data. And what you'll first see here is that these images look and actually are qualitatively very similar to those cartoon drawings that I showed earlier of the space of all particle images, except now, instead of looking at this extremely high dimensional and impossible space, we're looking at a very small 4-dimensional subspace where every particle image is laid out neatly, and we can see how they're arranged, how the topology and geometry of that space is arranged, and what the relationship between different particles or their clusters are. So we can see that in the variability component 1 versus 2 scatter plot, there are some clusters, there's some clear spread along one of the directions and even the other direction in terms of just a continuous spread of particles that exist from one end to the other. Same with variability component 2 and 3 and 3 and 4, we can see different patterns arise. And the components themselves, variability component 1, for example, here at the bottom, this image is showing in red a version of the 3D structure with the base density minus the variability component, and then the base density plus the variability component in blue. So we can see what kind of variability is being tracked here. And the first component, the main thing that happens between one side to the other is that the head region of the 40S subunit goes missing. So it dissociates or is broken off or in some other way is not present on one side of the variability. Variability component number 2 shows a ratcheting motion of the 40S subunit. This is now looking from a bottom-up direction. I'm going to show some videos of these same components and 2 more components so we can see what they look like. This is variability component 1. And so this is showing the head region of the small subunit appearing and disappearing along with some what looks like coordinated motion of other regions of the molecule, of the ribosome. Show that again. Now the second component is a ratcheting motion of the 40S subunit. And you can see it rotating here in the image. What's important to recognize here is that these 2 different motions or 2 different heterogeneous changes are being solved simultaneously from the same particle images at the same time. There's no classification here, and every particle image is actually allowed to have a continuous amount of variability component 1 and variability component 2 jointly. And so any single particle image can have any amount of either of these components. Same with component 3, which in this case is a different motion of the 40S subunit, this time just shifting left and right by a small amount. And then component number 4 is a rotation of the head region of the 40S subunit. So all of these motions, what we can infer now from the results here is that all of these motions and all these flexible continuous changes, other than the disappearance of the head region, actually happen simultaneously. And different particles may be in a different position along any of these axes, either with the head region not there or with the head region rotated or any other combination of the ratcheting and other motions. Every particle image may be at any position along these dimensions. So now that we have a feel for what 3D variability components are and what the latent coordinates represent, we can dive into some more interesting properties of the algorithm itself that make it useful in practice and make it easy to interpret and understand the results. The first and one of the most important properties is that the algorithm that we've used under the hood to solve the 3D variability problem is a member of what's called the Krylov subspace family. Krylov subspace methods are a set of techniques from linear algebra and optimization that operate in a certain way and have some theoretical guarantees. And one of the very interesting properties of cryo-lab subspace methods, which I won't get into in terms of their definition or anything, or the proofs, is that when they're solving a problem like this, in general, an eigenvector problem, they will solve, these methods will always solve variability components, in our case, in order of their variance. So that means the first components to be solved will be the most important in terms of how much variability within the data set they explain. If anyone's familiar with PCA, principal component analysis, it's also a similar method and it has similar properties under the hood, that it will, if you take the most important principal components, those will explain the most variability in a data set. Similarly, with 3D variability analysis, the first components will explain the most variability. What this means in practice is if you run 3D variability analysis with 3 components, the algorithm will solve 3 components, and then those can be sorted by their variance, since we can measure that, and we'll know which is the most important component. If you run the algorithm again with 6 components instead of 3, you'll actually get the first 3 components back again, because those are the most important, and so they're part of the subset of 6 as well, and you'll get 3 more components that have lower variance, meaning they explain less variability than the first 3. And you'll get this monotonously decreasing or equal variance set of components that describe different types of variability in the data set. In practice, we often run 3D variability analysis between 3 and 10 variability components or more, depending on how much GPU memory you have and the box size of your particle. And it's important to keep in mind that there is numerical precision to account for and other things. And the first few components can often have a very similar variance, so they may not always appear in exactly the same order. But if you look at the first 3 or the first 6 or so, they will contain the same variability components as a subset if you run it with more components. So that's a reassuring fact about the algorithm, that it will find the most important details first. The second important property is that 3D variability analysis is not a simple optimization problem, like the actual optimization problem that we're solving, this maximum over probabilities, is not simple, and it's actually not convex, but it's been proven that for probabilistic PCA type problems, the type of algorithm that we're using, that while the solution does have multiple local optima, all of them are equivalent. What that means is that when we're actually doing this optimization problem, trying to find the 3D variability components that fit the data, there are many different possible solutions, and some are good and some are bad. And we're measuring their fitness by their likelihood of explaining the data that we saw in the microscope. It turns out there are multiple locally optimal solutions. And so that's kind of disconcerting. You would think, if there are multiple solutions, how do you know which one is right? It turns out that they're all equivalent. They have the same likelihood, and they're actually all the same because they're related by a symmetry, which is that you can, if you're solving 4 components, you can switch the order of any of those 4 components while you're running the optimization and get the same result. At the end of the day, you can sort by variance, and you'll end up with exactly the same result. And that's another useful property of this type of algorithm, which is that the results that you get don't depend on initialization. It doesn't matter how you started off. It doesn't matter what your initial guess was. In fact, we don't need an initial guess at all, because any optimization from any starting point will yield an equivalent solution. And so that's another reassuring thing about the algorithm, that we don't need to worry about prior knowledge. We don't need to worry about initialization bias or tuning any parameters. And most importantly, we can't actually miss anything. Unlike 3D classification, where from run to run, each time you run it, the clustering may yield a different set of clusters. You may find some subclusters one time, not another time. You may have to run it again, and the number of components, the number of clusters that you run with will totally change the result. 3D variability doesn't have any of those problems. It has instead these properties that mean that it always finds the same variability components in order of importance, and it can't miss anything because all solutions are equivalent. There are a few other properties that are more sort of important in real life as well to discuss. One of them is that it's interesting and useful sometimes to think of the results of 3D variability analysis in terms of the spread of particles in the latent space as being connected to energy of the conformation of the protein. And that connection is there in the sense that we can measure the probability of a protein being in a certain conformation by assuming that the number of protein particles imaged that fell into a certain region of latent space represent the probability of the protein being in that conformation, which is then connected to the energy of that conformation via statistical physics. However, that connection we're not at the point where we can make that connection in a quantitative manner. And so if anybody's using 3D variability analysis, I wouldn't encourage drawing conclusions in an energy related sense yet. Detectability is another important property. It turns out that with 3D variability analysis, and we'll see this in a couple of results, small changes can be detected, much smaller changes than you would expect from, for example, the rule of thumb that proteins below 100 kilodaltons or subunits below 150 kilodaltons, if you're doing a local refinement and so on, can't be aligned. Those limitations no longer apply to 3D variability analysis because small changes can be detected as long as those changes are coordinated so that enough mass is moving in any which way, in any direction, and rigid or not, flexible or not. A coordinated motion or change of mass density, rather, in the structure will cause a detectable change in the particle images, will cause a detectable variance amongst particle images. And so much smaller changes can be detected than what would previously otherwise have been thought in terms of a rule of thumb. And that motion, like I said, doesn't have to be rigid. Finally, it's very, very important to remember that 3D variability analysis, because of its construction, will always return variability components that are uncorrelated, in the sense that if you compute the correlation between the latent coordinates of particle images, the correlation between two variability components will be 0. But that does not mean that the variability components, the heterogeneity that each one is capturing, is actually independent. Independence would require that knowing something about one of the variability components, a particle's position along one of them, doesn't tell you anything about the other. But you can see, for example, in this small example, knowing that variability component 2 is high or low tells you a whole lot about variability component 1. And so we don't have independence. And in general, it's not safe to assume that just because 3D variability found 2 different motions as 2 orthogonal variability components, that those motions don't happen together, or that there's no connection between those motions in terms of a mechanistic or functional sense. So that's also important to keep in mind. Next, I'm going to go through a set of practical considerations at the same time as showing some results on real data sets where those practical considerations were important, so that we can kind of discuss all these various points in a way that is contextually making sense. The first 3 together are probably the most important aspects to keep in mind when you're using 3D variability analysis in practice. The low pass resolution limit that you can set as a parameter when you're running the job in cryoSPARC controls the resolution to which we solve variability components. And this is tightly connected to the property that I mentioned earlier, that 3D variability analysis represents motion depending on the size of the object. So if we're looking at an object that has very large motion, we need to limit the resolution to a low resolution so that the shape of the object is large and doesn't have small details. And that way, the motion of that large object can be well represented by 3D variability analysis. On the other hand, if we're looking at a small object that is moving by a small distance, for example, if we're looking at the motion of alpha helices or anything at that scale, we need to set our low pass resolution to a high resolution, let's say 4 angstroms, that allows the algorithm to actually detect changes at that high resolution. So that's probably the most important parameter that modulates the results you'll get. The second parameter that's important is a high pass resolution. This kind of has the opposite meaning. It means that the algorithm will ignore low resolution information below a certain resolution. And this is important to get rid of variability that's caused by things that you don't care about. For example, with membrane proteins, a lot of times you may have detergent floating around, empty micelles, other particles, broken protein, denatured stuff floating above or below the particles in the ice. And that variability from that extra mass can often swamp the variability caused by the motion of the protein itself. And so using a high pass resolution when you're not seeing things that look like motion, if you're seeing variability components that just have large blobs moving in a way that seems to be disconnected from the actual protein, then applying a high pass resolution can often help. Finally, masking is probably the most important thing. It allows you, the way the algorithm is built, supplying a mask allows you to include or exclude regions where the variability that may be present there in terms of the real space structure of the protein is not biologically important. So a perfect example of this is with membrane proteins where you don't really care about what's happening in the micelle. So for example, this first example I'm going to show is a GPCR complex, a cannabinoid GPCR complex, a public data set that's available to download. I think this has 300,000 particles that were used for 3D variability analysis. The actual consensus refinement structure of the GPCR shows the membrane, the protein itself, multiple subunits of the complex, the transmembrane domain, and the detergent micelle. Now, when we run 3D variability analysis on this data directly, what we end up finding are variability components that look like this. So the first component has, on the negative side, density appearing on one side of the micelle, and on the positive side, density appearing on the other side of the micelle. And in between, the density moves from one side to the other. Variability component 2 is similar, but just in a different direction. And so all we're finding here with 3D variability analysis are the types of change and the differences from particle to particle in the shape of the micelle. That's interesting, but not really what we are looking for. So what we can do is create a mask, a soft shaped mask that only contains our protein, excludes the micelle, and now run 3D variability analysis inside this mask. And it's only going to look at variability inside the mask. What we end up with there are some results that are really nice. Even for this small membrane protein, we can see directly continuous flexible motion of the actual complex. And the first component that we see is a clear bending mode where the transmembrane domain and the soluble parts move closer and further apart. And various different parts of the protein are moving not in a rigid way, but in a flexible bending motion. And because of the small motion here, we can use 3D variability analysis at a resolution set to 4 angstroms. And so we're able to see detailed motion even of helices and helical pitch showing up as we move through this motion. And the video here that we're seeing, I should have explained earlier, is created by taking 20 snapshots at positions along the variability component, generating a volume from each one, and then displaying those as a movie. So that's the first variability component. The second variability component shows us a different kind of bending of the transmembrane domain and the rest of the protein, this time with the transmembrane domain moving in a transverse direction from left to right in this rear view. But what we also see that's really curious is that there is a motion of a much smaller part of the protein, this 2 helix bundle here, that moves closer and further away from the rest of the protein in a coordinated way with the rest of the motion. And that's what's important. Even though this moving part of the protein here, like if you were to think of this, the largest rigid part here that's moving is very small, it's moving in a coordinated fashion with other parts of the protein, and so that motion is detectable. Component 3 is also very interesting. It shows us an independent, we don't know if it's independent, but a twisting motion of the transmembrane domain in a different direction. And this time there isn't much else going on in the rest of the protein. And that's interesting because this part of the protein that's moving here is less than 50 kilodaltons. And we're able to detect that change even in this small protein, even inside of the micelle. I'm just going to play all 3 of these videos for you here right now. Great. Okay. The next practical consideration and set of results we're going to talk about is strategy around subclassification and doing variability analysis when there are multiple different levels of heterogeneity present in a data set. The data set we're going to look at is a flexible membrane protein, an interesting protein that's therapeutically relevant, NaV1.7 channel, which is a pain receptor. The original publication and original data upload was done just a few months ago last year. It's a very large data set with 25,000 movies, and the original publication was able to find, by standard classification methods, an inactive and active state of the ion channel. There are also 2 Fabs bound to the structure, and there's spider toxin bound. And what we were able to do in cryoSPARC was actually substantially improve the resolution compared to the published structure using non-uniform refinement due to the disordered micelle. And that's an important note because 3D variability depends on the 3D structure and poses that come from a consensus refinement that was done ahead of time. And so being able to use a better refinement technique to get a higher resolution structure globally, even despite the flexibility in motion, is actually helpful for 3D variability analysis. So with this heterogeneity, we already know that there's a discrete conformational change between the open and closed versions of the channel. And that's actually easy to separate with standard techniques because the 2 versions of the channel are substantially different and the particles exist in one conformation or the other. So that can be done with regular techniques. And this is what we would suggest typically when you have a data set of any kind. Use the standard techniques first to try and figure out the different discrete conformational changes that are present. You can also use 3D variability with the whole set of particle images, and that'll show you what kind of conformational changes are present, and then you can cluster the data set into subclusters, and then apply 3D variability analysis again to each of those subclusters. So in this case, what we did is take the active state only and apply 3D variability analysis again. And what we were able to find are very interesting types of motion that happen within one of the already 3D classified subclasses. The first component shows bending of the two fabs closer and farther apart. And those two fabs are bound tightly to two of the subunits. The protein is actually fourfold symmetric, but there's only space for two fabs. So the two fabs are bound to two of the subunits, and it's actually the bending of the transmembrane helices and their motion by several angstroms that allows the fabs to bend. And that motion explains why the resolution of the structure in the peripheral regions of the protein is so poor in the reconstructions that we have so far. The second, we actually solved 6 components, and they all show different kinds of motion, but I'm just showing a selection here. The 6th component shows bending of the other 2 domains of transmembrane domains in the same way through the membrane, which also explains the poor reconstruction quality there and suggests that the entire transmembrane region of the protein is quite flexible. This is just all 3 different components together, and the middle one shows bending of the 4 helix bundle in 2 different directions. I'm going to skip this to move on just in the interest of time. The next thing I'm going to talk about briefly is symmetry and symmetry breaking. It's well known that symmetric molecules are only symmetric up to a certain resolution, and at some point, heterogeneity, rotamers of side chains, and so on can break the symmetry. What we did is apply 3D variability analysis to a standard data set of T20S proteasome images, which are thought to be very rigid and used as a test sample for single particle microscope testing. What we found with 5 different components and no symmetry enforced is that we were able to actually find different kinds of bending motion within the complex. And the most interesting thing was that 3 of the modes that we found were symmetric, meaning that the bending itself was symmetric in the same way as the protein. But 2 of them were asymmetric, and these were bending types that broke the symmetry of the molecule. And so the first component I'll just show you is an extension motion of the barrel, where all the subunits are doing the same thing, all extending and retracting in sync. The second component is twisting of the barrel, again in sync, all the subunits doing the same thing at the same time. And you can see the motion of individual alpha helices here as well by a few angstroms. And this 5th component is a bending of the whole molecule across its symmetry axis. And here, each subunit is doing something different depending on its position within the complex. And so this is a very interesting way to see for symmetric proteins whether or not the symmetry is really there, or flexibility actually breaks the symmetry despite the chemical symmetry being present. Next, I'm just going to briefly talk about what to do when you have very large motion, where, for example, with this precatalytic spliceosome complex, 3D variability with 2 components solves for 2 different kinds of very large motion of the helicase and head regions of the protein. In this case, we can look at particle images that have similar latent variable values, cluster them together in the latent variable space, and perform local reconstructions of just those particles in a moving window fashion across the latent variable. Instead of just looking at the variability component, we can do reconstructions of particles. And this is a common technique for cryo-EM manifold embedding methods. What we see there is that we can get higher quality intermediate reconstructions that show us motion in a more interesting way. And this is for the precatalytic spliceosome, one of the components. And the other component, moving in a different joint manner of the 2 moving pieces, along with transverse motion of the foot region. Showing those 2. And finally, I'll just talk about one last thing, which is that in some cases, you can actually use the latent variables that come out of 3D variability analysis to cluster particles into discrete classes as well. So for that, I'm just going to switch over to cryoSPARC itself. This is a result that was computed on a data set of bacterial large ribosomal subunit molecules, which form different assemblies, intermediates, in an assembly pathway. And what we can see, this is a 3D plot now of variability components that are showing the three different components and the location of particle images along those components. Each dot is a single particle image, and they're colored by clustering that we did after solving the 3D variability components. Each colored region corresponds to one cluster, and it turns out that those regions, which are well separated in terms of the 3D variability space, actually correspond to, this is the same plots, the same scatter plots of the 3 dimensions, as well as colored versions of those with the clustering, and some unwrapped versions so we can see the clustering in a more efficient manner. Those clusters actually correspond to different discrete versions of the ribosome protein. The green cluster is a small version. I don't know exactly what all the different conformations are called, but you can see the different conformations being generated as we do reconstructions just of particles from within each cluster. Right, so that's an example of doing clustering using 3D variability analysis, and all that is supported right within cryoSPARC. Finally, I'm just going to show a couple of examples of recent uses of 3D variability analysis in structural studies that were published just in the last few months. The first one is a SARS coronavirus 2 spike protein. And this was the first paper showing the structure of the coronavirus spike protein. And they were able to use 3D variability analysis to discern flexible motion of the whole spike protein, and especially motion of the receptor binding domain that was in the up position, as well as motion of this sort of tail region here. These are 2 different views of the same variability component that we're seeing here. The next example is an even more recent example, a structure solved and 3D variability analysis used for the GABA B protein, just published a few days ago in Nature. And here they were using, the team was able to use intermediate reconstructions to solve 5 different conformational positions of the GABA protein bending along its length. Okay, and that's actually the end. And just to conclude, 3D variability at this point is a first step in our pathway to solving heterogeneity really well for single particle cryo-EM and getting to that final state where we can really resolve all the different conformations and high resolution structures, even of flexible proteins. It's effective already in making it possible to investigate protein dynamics straight from the image data without relying on simulation or prediction. And it can resolve and expose biologically and functionally useful dynamics and mechanisms that people are finding already in their work. So I hope that everyone who's listening today has a chance to try 3D variability analysis on your own data and see what happens. And always, of course, please feel free to contact us and ask questions on the discussion forum. And other users who are using 3D variability analysis will also be able to chime in to give their thoughts. We're hoping that with 3D variability working as well as it does, it's giving us some insight into how a next version of fully flexible refinement could possibly work. So unfortunately, I'm a little bit over time, but I'm going to stop there for questions.
Perfect. Thanks so much, Ali. So we'll go, we have reached the end of the hour, but we will stay on for question period, and for anyone else who'd like to stay on for that. We'd also really like to get your feedback, as this is the first webinar that we offer, and we hope to offer more in the future. So I'm just launching that poll right now. It's very simple, 3 questions. They're all anonymous. If you can take a moment to complete that, we would very much appreciate that. And we'll go right into questions. So I've tried to organize these into a little bit of a chronological manner. So starting off with 3D variability in terms of its theoretical foundations. Let's say we were to just take the 2 extreme volumes from an individual variability component and use the morph map function in Chimera. Will we obtain a similar result as to what you've shown here for 3D variability?
You would obtain, so depending on how you create the movies, actually the tutorials that we have online explain a few different ways to do it. Depending on how you create the movies themselves and how volumes, intermediate volumes are thresholded, you may or may not get exactly the same thing. But theoretically, the linear subspace model is linear. And so if you have the 2 endpoints of the variability component, you can generate every volume in between by doing a linear interpolation between those. So you should end up with something that's probably similar to how morph map in Chimera works, yeah. But solving the endpoints is just as hard as solving the variability components themselves.
Thanks. Going back to our linear subspace model, you had mentioned how the X term is our 2D image term. So just for clarification, does the XI refer to one particle, i.e. one view within a micrograph, or does it refer to the entire micrograph that contains several particles?
Great question. Each xi is a single particle image. So there's just one particle in there, and the I is the index for which particle we're talking about. So you could have multiple x's within a single micrograph for sure.
Perfect. So moving on to talk a little bit about variability components themselves. You mentioned that we have positive and negative values. Do those compensate each other to create the 3D volume that we're looking at?
Yes, exactly. So any density, when we look at the 3D variability components themselves, let's just get to a picture here. When we look at the 3D variability components themselves, any positive density in V0, the basic density, can be offset by negative values in the variability component, and any empty spaces can be filled in by positive density in the variability component, or vice versa. And you can add density or subtract density from anywhere. So they could definitely offset in different directions to create the variability that we see in each of these images.
Perfect. Are all of the variability components generated from the same data set? So I guess the question here is, are they generated from an individual data set, or are we combining particles from different data sets to find all of the variability components?
Great question. No, in this case, every result that I've shown and that anyone that I know of has done, all the images come from the same data set, usually the same grid, same sample, and same set of images. And that's actually very important because if you try to combine images from different data sets, for example, different data collection runs, different days, there may be other kinds of variability between those particles that end up getting picked up by 3D variability analysis instead of the protein variability, which is what you're interested in. So for example, if the ice is much thicker on one day or another, or your defocus settings are very far off one day or another, or the magnification is a little bit different, those changes will end up getting picked up instead of what you're really interested in.
Thank you. You mentioned that the linear subspace model is related to the Krylov subspace models family. And you also mentioned that in that set of models, the components that are first picked up are the most important. Can you elaborate? Does one know at the beginning what are the most important components? And if so, how? And I guess here we need to make a connection between what the model can observe versus what we as humans and scientists want to observe.
Right, exactly. So before running the algorithm, we don't know what any of the components are or their importance in the sense of the amount of variance that they explain in the dataset. The variance itself is actually very simple. We can literally take since each of the variability components, like the actual VKs in the math, I'll just switch back to that, are normalized, this equation right here, each VK has the same amount of information. It contains the same amount of change of the density. And so we can literally look at the spread of particle images along the variability components in terms of their latent variables and say, okay, variability component 1 spreads between minus 500 and plus 500 units of change. And variability component 2 spreads between minus 300 and plus 700 units of change. That's the amount of variance. And in fact, you wouldn't just measure the range, you would literally measure the variance. That's the importance of a component. And so we don't know that ahead of time. We don't know what the components are, but the nice property of this algorithm is that it will solve the components in the order of their variance. So it will pick up the high variance components first. If you ask for 3, it'll get the top 3. They may not be in order of variance when they actually come out of the algorithm, but they will be the top 3. And then in the code, we actually measure the variance and sort them by the variance. So that ends up putting them in the right order anyway. So hopefully that answers the question.
Perfect, thank you. So I'll move on to some of the more practical types of questions now. So starting off with validation, is there a way that we can quantitatively know that the variability components that we are observing through the linear subspace model are in fact there, or in fact can be relied upon?
That's a great question. Because 3D variability analysis, because the algorithm has these strong theoretical properties, it is much more trustable than, for example, running 3D classification and wondering whether a particular class exists or not. And because we can literally see or rather with 3D classification, wondering whether a class is intact or whether there's still subclasses and so on. With 3D variability analysis, because of the strong guarantees, and because we can see the spread of particles along each variability component and then see what those variability components are, the feeling of trust is there. However, we don't right now have a quantitative or theoretically verified way of validating 3D variability results in the sense of like something like a Fourier shell correlation. something like that could be possible in theory, and that's something we're going to work on next. But so far, the dynamics of the proteins that are resolved often corroborate very well with other experiments, and that's usually a telling enough sign for the studies that we've seen being done so far.
Great. Is there a requirement for a certain number of particles in order to reliably carry out 3D variability analysis?
For sure. So there isn't a strict number, but you do need a fairly large number of particles. So I haven't seen any good results coming from data sets with less than 100,000 particles. And this doesn't have to do with the resolution. Normally, when you're doing reconstructions in cryo-EM, the number of particles kind of relates in a log linear way to the resolution. With 3D variability, it's not about resolution, it's about sampling of orientation space. To do 3D variability analysis properly, you need to have seen the protein from the same view multiple times, but at different positions of the latent coordinate. So you need to have seen different versions of the protein. Let's say with the ribosome, you have the twisting of the small subunit. You need to see the bottom view at different positions of that small subunit's motion. So what happens if you don't have enough particles is that you just don't have enough sampling to have seen the same object from the same direction, but with different conformational positions. And so orientation sampling is actually really important. If you have a highly skewed orientation distribution, you need more particles. But generally 100,000 or more is probably the lowest, the minimum that you need.
Thank you. Relatedly, how would the size of my data set or the number of particles affect the number of components that I might initially choose to start off with?
Great. So the size of the data set will actually be surprisingly not related to the number of components that you can solve. It will be related to how, it'll be related in the sense that if you don't have enough particles, you won't be able to solve any components well, or you won't see all the different motions since they won't be well sampled. But the crux of the matter is that for 3D variability analysis to work, what we're doing is adding more degrees of freedom to the problem on a per particle basis. So every single particle image has to have enough signal to lock down all of the ZIs along with all of the poses that were also unknown. Since the poses are unknown for every particle image, and now the ZIs are also unknown for every particle image. So when you add more particles to the data set, you don't actually add more statistical power to be able to infer the ZIs for a particular image, since only the data from that particular image can tell you which position of the motion is that particular image in. And so the size of the molecule helps a lot. A larger molecule will have more contrast, and the thinness of the ice also matters. But other than that, the size of the data set will be less relevant for how many different components you can see, at least in our experience so far.
Thanks. Would 3D variability analysis be helpful in showing or assessing preferred orientation problems?
It definitely will suffer, and probably worse than other methods like just standard refinement from orientation issues. I'm not sure if there will be a way to diagnose orientation problems from 3D variability analysis other than just seeing that 3D variability analysis doesn't work. It solves components that are clearly not having any relationship to the 3D structure of the protein. Normally when you have missing views, what you'll see in your 3D variability components is streaks of positive and negative density all in the same direction, which are corresponding to the directions where you have missing views. And so you can tell that something's wrong, but it won't really help you to do anything else.
Going back a little bit to what we can actually infer from the results of 3D variability analysis. You had mentioned earlier that the observations that we can make, we can't necessarily connect those with a particular underlying assumption about what's happening. So is it possible that some of the variability components come from vibrations of molecules in the ice rather than intrinsic conformational variance in the protein itself?
I'm going to guess that the question is about thermal vibration or like thermal motion.
I think so.
Yeah, I'm not too sure in terms of the sample preparation and the amount of motion that is plausible at the temperatures where the sample is stored, like in terms of motion of a given particle while it's being imaged. I'm not sure about that, but thermal motion of the particles is probably definitely responsible for all the variability that we see. And if my understanding is correct for the statistical physics side of it, the molecule itself has an energy landscape that if it's not a perfectly rigid molecule, the energy landscape is somewhat shallow. there are multiple different shapes or conformations, positions of all the atoms that are energetically not too far apart. And at a certain temperature, like at room temperature before freezing, the particle can randomly, just by Brownian motion, sample that set of energetic positions or low enough energy positions. And then when it gets frozen into one of those positions. And that's why there is a link between how many particles you see at every position along the variability components and the energy of that position and the statistical likelihood that the particle would ever be in that position because of that Brownian motion, the thermal energy that makes that random sampling happen. So it's definitely driven by that as far as my understanding goes. And of course, there can be other kinds of changes that have other dynamics as well. Like if you have a ligand binding or some other kind of sort of irreversible event, you may have, or equilibrium process, you may have some particles that are on one side of it or the other, and that may also be a continuous process. But it's almost all driven thermally. Hopefully that answers the question.
Perfect. Okay, going a little bit now into the case of heterogeneity. Let's say we can see two distinct states already in our data set using heterogeneous refinement or other 3D classification techniques. Right. What is the best way to search for the 3D continuous movement between those states using 3D variability? So for example, should we separate out the two states and then perform 3D variability analysis on each one separately, or should we do this as a consensus structure?
That's a good question. The answer is actually that you shouldn't try to do that, because if you are seeing and you're convinced that you have discrete populations, then there actually isn't data in the sample to support any conclusion about what happens in between. Since we don't actually see any particles in between, we see particles at one end and at the other end. And so we can't really infer exactly what happens to the protein in between those two states. And I would say that if you tried to make a conclusion out of that, it would be hard to hold up because we don't see those particles. In terms of seeing what happens to the particles in one state or the other in terms of the thermal motion they're able to undergo in that state, then you should split the data set by standard classification methods, or even using 3D variability analysis at the top level with all the particles, and then split it based on clustering the latent space if you see clusters there, and then do 3D variability analysis again on the subsets. Then you'll be able to see what kind of motion there is within each of the discrete classes, just like I showed with the nav channel. But you shouldn't try, just like in this case, it wouldn't be right to try and guess what is the pathway of motion between the active and deactive state, because we don't really see particles in between that.
Great, thank you. Moving on to some more practical questions about actually using 3D variability analysis in cryoSPARC. You mentioned that we need a consensus refinement to start off. So the question is, why doesn't 3DVA ask for the consensus volume, but rather for a mask as the input to this job?
So when you're actually using 3D variability analysis in cryoSPARC, it does require a volume input. No, actually, no, you're right. Sorry, it doesn't require a volume input.
Labeled as mask.
Yeah, it requires the particles input and a mask input. The reason is that the particles have to have their alignments. And if the particles already have their alignments, we can actually do a reconstruction with all the particles without having the initial volume to start with, or the volume from refinement to start with. So it's actually easier and cleaner. And the way it's implemented is that you connect all the particles and the particles have their alignments. The first thing 3D variability does is reconstruct the consensus refinement from all the particles and their fixed alignments, and then carry on with 3D variability analysis with the mask that you provided.
Thanks. How should we actually go about working with the different, I believe there are different modes, right? So there are different modes implemented in the 3D variability analysis job. Should we start off with a simple mode, or is there a particular approach?
So the different modes they're actually in the 3D variability analysis display job that helps you create the movies and other display items coming out after 3D variability analysis. 3D variability analysis is computationally heavy. It's actually quite fast compared to relative to how much work it's doing, but it does take probably a couple hours or a few hours on a large data set on a single GPU. And so we don't want to run it over and over again, but with the same results out of one 3D variability analysis run, you can do different actions to create outputs. So the display job has simple mode, cluster mode, and intermediate mode. Simple mode just creates this simple movie of 20 frames or however many frames you want, linearly interpolating along the linear subspace model to show you what each variability component is capturing. It doesn't take into account anything about the distribution of particles in the latent space or clusters or anything like that, just simple. The cluster mode does what it says, which is it takes the particles in the latent space, clusters them into as many clusters as you ask for using a simple Gaussian mixture model clustering, and then reconstructs a 3D structure from each cluster and outputs those. So for example, what I showed with this ribosome intermediates, each one of these blue maps here is an independent reconstruction from particles that were just like, for example, this is the green cluster. So only the particles that were in this green cluster are used for this reconstruction. And that's automatically done if you choose the cluster mode, and you have to provide the number of clusters that you want to do. And the intermediate mode does the last thing which I showed here, which is create intermediate reconstructions by locally weighting per particle latent variables so that we can figure out which particles are similar to each other in terms of their conformation and just do a reconstruction from those. So the output of the intermediates mode will be videos like this, which are not linear anymore. And so like for example, the previous question, these videos would be very different from what you might get from Morph Map and Chimera with the endpoints, because these ones actually do reconstructions of intermediate particles along the way. So those are the three modes, and you can decide, depending on what you're doing, which one you'd rather use.
Thanks. Some more notes on parameters that we need to set. You mentioned that we can set a high pass filter and a low pass filter resolution. Is it in practice, should we try applying different values to those parameters to visualize different types and sizes of motion?
Sure. So I would say as a general strategy, you should start with a lower resolution and run 3D variability analysis, see what you get. And if you see motion at a low resolution, I mean, small motion may not show up, but any motion that you do see, if it's small relative to the size of the low pass resolution that you set, you should increase the resolution and try again. In fact, you should just run them in parallel if you have the GPUs, do like 10 angstroms, 5 angstroms, 3 angstroms, depending on what resolution you got to in your consensus refinement. It doesn't really make sense to do a 3 angstrom 3D variability when you're at 8 angstroms in your overall refinement. So the 3D variability refinement, sorry, the 3D variability low pass resolution should be set to a lower resolution than what you've refined to, but otherwise you should try different levels for sure.
Is there a particular resolution that our consensus refinement needs to have achieved before the results of 3D variability will actually show us something that we're interested in?
Not strictly speaking, but probably there is relative to the size of the molecule. So if it's a really large thing like a ribosome and you get to 10 angstroms, then 3D variability will just work fine and show you motion, because you're not really interested in motion smaller than 10 angstroms with something that's that big and moving by large amounts. If it's a tiny membrane protein and you're at 10 angstroms, then 3D variability is not going to do anything, because there aren't even any features that you could see moving or really any interpretable thing that you could get out of it, since you can't actually identify alpha helices or any structural elements from which you could derive some insight. So I would say that's kind of probably the rule of thumb.
Perfect. Can the variability component values, once you've obtained them using the 3D VA job, can they be exported from cryoSPARC and then replotted elsewhere?
Yeah, for sure. The variability component values are all outputted in a cryoSPARC file that is the output of the 3D variability analysis job. I believe we have a tutorial online about how to read the cryoSPARC file format. It's actually a very simple NumPy file. So you can, if you have Python and NumPy, you can just open up that file and see all the rows and columns of, it's a tabular data format. And there's columns that are called component 0/value, component 1/value. And those are all the variability latent coordinate values of each particle for each component.
What can we do with the output of 3D variability analysis? Can we somehow use that to further improve our refinements, or is there anything that can be done?
There's one obvious way, which is that if you see motion, like for example, in this spliceosome example, when you're seeing this kind of result, you definitely know the next thing that you should try is a local refinement or multi-body refinement or something like that of these pieces that seem to be moving in a more or less rigid manner. And because of this 3D variability analysis result, you know which parts of the protein are moving together and which parts are kind of at a hinge point and so on. So creating masks and doing local refinements is a great next step, but that only works if your particle is big enough to begin with that you could cut it up into pieces and the motion is actually rigid. If those aren't true, then you can't really do too much in terms of improving the resolution yet. I think that's kind of like the dream of doing heterogeneous flexible refinement, is that now that we can see the motion, why can't we undo it? So that'll be the next frontier.
Perfect. I see that we're at about 12:20. Perhaps there are still quite a few questions. We can perhaps go for another 10 minutes and then close off the Q&A.
Sure.
But we would like to very much thank everyone for participating and attending this webinar. The recording will be made available afterwards online, and you'll all be informed of that. If you haven't had a chance to fill out the poll yet, please do so. We'd really like to have your feedback because we do want to keep doing more of these. And we'll just spend the next 8 minutes or so on some more practical questions that are coming in.
Great.
Thanks. All right, you kind of alluded to this already, but can you tell us a little bit about how 3D variability fits in a standard workflow? So for example, what would you want to have done before? What can you do after? You mentioned local refinement as a way to observe more smaller regions.
Sure. So the pretty much template standard workflow that anybody who's doing this for the first time should follow is get your particles, get your data, do all the pre-processing, do some particle picking, do a bunch of 2D classification, and try to get rid of as much junk as possible, like obvious junk, but don't go out of your way to remove things that could be particles, or things that definitely are particles, but maybe a different conformation or something like that. Don't try to separate those at the 2D classification stage. Just get rid of any obvious junk. Junk doesn't work, or 3D variability analysis doesn't like junk too much because it's such a strong variability that many of the first variability components will be swamped by just junk shapes that are misaligned particles. So you don't want that. So get rid of as much junk as you can at 2D classification. And even if you're doing multi-class ab-initio reconstruction or something like that, try to get as many clean classes as you can, and then combine all the good particles that you have from any type of input, all the good particles into one particle stack that is connected to the input of a single 3D variability analysis job. Maybe try that with 3 or 5 variability components so it doesn't take too long. And set it to, depending on what resolution, you have to take all those particles and do a refinement first. That shouldn't take very long. Depending on what resolution that gets to, set your low pass resolution for 3D variability analysis based on that, and run 3D variability analysis with 3 or 5 components. What you should see is that if you saw any heterogeneity earlier and you recombined all the particles, that heterogeneity should resurface after 3D variability analysis in terms of either discrete or continuous changes. And you'll be able to see in a lot more detail exactly what's happening in your molecule. And then try to do either from there, guided by what you've seen in 3D variability analysis, do some heterogeneous refinement, or use the cluster mode of 3D variability analysis display to separate your data set into different clusters that are ideally relatively different conformations. And then for each cluster, do 3D variability analysis again to see what kind of continuous changes are left after that. That would be kind of the standard exploratory workflow. Once you've seen that, once you've run through variability analysis once, you'll be able to see pretty quickly that, okay, I'm seeing motion, but the motion's not that large, so I can probably try with a higher resolution, get more detail about the motion, or vice versa. And you'll also be able to see if you have regions of the molecule that are contributing variability that you don't care about. So maybe there's a part of the protein that's always floppy and disordered. You don't care about that. You want to mask it out, create a mask at that point based on the results so far, run 3D variability analysis again. That's kind of like the general standard workflow. And then after that, creating local refinements would be another possible next step, or just doing more subclassification and then doing regular refinements of each different subclass that you get to try and push the resolution there.
Perfect. In terms of masking, in the T20S example, the T20S proteasome example, did you assess the variability in symmetry breakage by applying the mask on the entire complex or only on the top part of the molecule? And more generally, can you speak a little bit about masking strategies when using 3D variability analysis?
Sure thing. So the symmetry case is actually interesting. Because I was trying to get through it a little quickly, I didn't mention all the details here, but the most important thing is that in order to get this result, I took all the particles, did a refinement, and then applied symmetry expansion to provide each particle, or allow each particle to represent every subunit that's in it, and then did 3D variability analysis using the expanded particles. And 3D variability analysis does not support any form of enforcing symmetry. So every particle, when you do symmetry expansion, now every single copy of every particle is allowed to have its own variability coordinates. And that's what allows every subunit to independently have enough degrees of freedom that you could find different subunits doing different things at the same time. In terms of masking strategy for that case, there are two options depending on what you're trying to do. If you want to investigate only intra-subunit variability, like what kind of thing happens inside of one subunit without regard to other subunits, then you should create a mask that only contains one subunit, do the symmetry expansion, and then do 3D variability analysis. You'll have lots of data because of the symmetry expansion, and you'll be able to see exactly what kind of detailed motions happen inside of one subunit. If you want to see how the symmetry breaks, the previous approach won't show you that. To see how the symmetry breaks, you have to use a mask that contains the whole molecule, but you can still apply symmetry expansion. The symmetry expansion in this case doesn't actually help, because when you do symmetry expansion and you have a symmetric object, I mean, without any other knowledge about it, you would expect that every variability mode that breaks the symmetry has to show up 7 times, or whatever the symmetry is, to be able to conform with the symmetry. Like if the T20S, for example, the barrel bends this way, it should also bend in every other symmetric way, since there isn't, as far as we know, any difference between the symmetric rotations of the structure. So the variability components should also have symmetric copies if you apply symmetry expansion. That can make things a little complicated, because then if your top component repeats 7 times, then you'd have to run 3D variability with so many components. So you don't have to do symmetry expansion, but this is actually still an evolving area. We're not entirely sure of exactly what to do always with symmetry. There's actually a bunch of text about it on our tutorials about 3D variability analysis. But masking, just to add a note about masking generally, you can do anything you want with masking, and the softness or sharpness of, or size or cutting through density doesn't matter too much. I mean, you should still use a soft mask whenever you can, but cutting through density doesn't matter, because we're not aligning the particles. So you can isolate any subregion that you want. However, if the region is too small, then just like other techniques, there won't be enough density, no matter what the variability is, there won't be enough density inside of the masked region to be detectable. And so you should try to mask the largest region you can that contains variability that you're interested in, including some parts that are not moving too much if possible.
Thank you. And I think the very last question here, is there a way to tell how much variance is captured by each component in 3D variability analysis, so that we can make a kind of a judgment about how much variance is explained?
That's a good question. We don't provide a quantitative version of that, like in terms of fraction of variance explained, because if you think about it from a theoretical perspective, the number of variability components that you could solve is actually only limited by the number of pixels in each image. And so that could be tens of thousands of variability components. And almost all of them will just be full of noise, and their variance will be small. Like if you think of all the variability components from the first one to the last one, the first ones will have very large variance that they're explaining. And then that variance will shrink, hopefully quickly, explaining all of the actual interesting things that are happening in the data set. And then all of the other components will be capturing the variability due to noise. And so at some point, the variability components end up being just noise. And we would ideally want to be able to detect that when the variance drops below a certain quantitative amount. We haven't got to that point of actually developing that threshold or figuring out how to compute that exactly right. So right now we don't provide any quantitative value of variance that we can give in the job, but you can compute the variance directly by just getting the variability components out of the CS file and literally computing their variance. You could also compute their range or their standard deviation or whatever else you want to do.
Great. Thank you so much. So as we've gone quite a bit over for our questions, I think it's a good place to end it there. Again, thank you to everyone who joined and stayed for this presentation. We really appreciate it, and we look forward to seeing you on future webinars.
Absolutely. Thank you so much.